So, in the last lecture, I did introduce to you the mechanism of immunoglobulin class switching. Today's class, we are going to deal with in-depth analysis of what goes on when immunoglobulin class switching takes place. Later on, I will also talk a little bit with regard to regulation of the immunoglobulin gene expression. Now, before I come to class switching, I would like to introduce the different classes of immunoglobulin, because this is relevant with respect to the constant domain genes which undergo class switching. There are 5 different classes of immunoglobulins: IgM, IgD, IgG, IgE and IgA. Now, when I say these 5 classes, we are referring to what is present in the mammals, human as well as mouse. Now, like I said earlier, that these are the 2 systems that have been studied in depth by immunologists, mouse and human, and what is true of mouse is also true of human. Now, why do I have the IgM in bracket? Is because always when the immune system responds to a particular pathogen or antigen, the first class of antibodies that are secreted in circulation are IgM. You might remember that B cells, naive B cells, have their B cell receptor as IgM as well as IgD. The 5 classes of immunoglobulins are as mentioned here: M, D, G, E and A. These refer to the association of the heavy chain. So, these immunoglobulins, the classes are called by the heavy chain. Of course, in case of IgM, the heavy chain is mu, D delta, G gamma, E eta and A alpha. The IgG has been divided into 4 different subclasses. Now, in my next lecture, I will be telling you all the structure and function of immunoglobulins, and you will have the opportunity to see how these 4 IgGs differ from each other. IgA on the other hand is IgA1 and IgA2. Now, these 2 subclasses are based on the fact that one exists as a monomer and the second one exists as a dimer. That again will be dealt with in the next class. I would like to reiterate here that except for IgD, the antigen receptor which is expressed simultaneous with IgM, except for IgD, antibodies corresponding to all the immunoglobulin classes and subclasses can be found in circulation in response to a particular immune response. I would like to again once more introduce to you constant versus variable regions on immunoglobulin. Now, the same thing also is seen as antigen receptor as well as antibodies. You have the variable region which has been chalked out here. This variable region is formed by the recombination that we discussed in the last class, VDJ recombination as happens in the heavy chain and VJ recombination that happens in the light chain. Now, needless to say, this variable region is the region which is required for antigen recognition, and variable suggests that each immunoglobulin is different in the type of sequence that it bears on the heavy and light chain at this terminus. 2/3 of the molecule remains constant, and therefore, this is called the constant region. All immunoglobulin, let us say IgG1, would be quite identical in its sequence in the constant domain, but very different at the variable domain. Now, before I come to again the class switching itself, let us see when does class switching take place. Remember, I have told you that class switching is a phenomenon that happens in case of B cells which are T cell dependent. I will remind you of certain molecules which are required for this class switching, but just let us look at the antibody titer that one can see in response to an antigen. What is written here on the y axis is the antibody titer, and x axis shows the days of immunization. Now, let us say this is a rabbit and has been injected on day 1 an antigen, and the animal is test bled every other day, and the serum obtained from this test bleed is then tested for the presence of antibodies specific to the Ig or the antigen. Remember that the total immunoglobulin repertoire will remain almost the same with respect to the amount of amount of immunoglobulin, but the antibody titer, the antibody to the antigen which has been injected, will differ. Now, what one can see very clearly that there is a lag phase of at least about 6 days, after which the circulating antibodies to the antigen start to increase, and the class of immunoglobulin that one sees in blue is mostly IgM. If one looks closely, one will be able to see that there is a very small population of antibodies that are of IgG class. This is much later than the IgM, and as has been seen via experiments, that class switching from IgM to IgG begins at around day 7 in the primary immunization. Now, when the same antigen is injected, after the circulating antibody titer comes down, then there is almost no lag phase, and there is generation of a robust immune response, and the antibodies that are present here would be IgG or any other isotype. Once again, you will see that there is a small amount of IgM which would represent those naive B cells which have been recruited in this particular immune response. So, you would have, now usually one would not look at these IgM, will not be able to detect these IgM in an immunoassay, because the affinity of this population would be much lower than this, and therefore, these are totally obscured. But nevertheless, there are IgM antibodies in the response here, which would be the primary response for with respect to some cells, naive cells that have been recruited. Nevertheless, let us just look at this. Now, again I would like to tell you that the affinity of these two populations also varies. This is high affinity compared to this, but what I would like to emphasize here, that the immune response that is generated in the primary would be mostly IgM, and there is generation of memory cells which is known to class switch to IgG type of antibodies, IgG class of antibodies, or any other class. Let us look at what are the other classes. Now, when we come to immunoglobulin class switching between the IgM heavy chain, which is called C mu, and C delta, because these always are co-expressed on naive cells, you can see that these can class switch from mu d to any one of the isotypes. What are these isotypes? They are the constant domains, remember, which we have seen in the immunoglobulin gene organization. This is exactly that, the immunoglobulin constant domain genes, which are always in this particular sequence: mu, delta, gamma 3, gamma 1, gamma 2b, gamma 2a, constant eta, constant alpha. Now, just like to remind you again that gamma in human it is gamma 1, 2, 3, 4, whereas in mice it is 1, 2a, 2b and 3. Now, the recombination, when naive cells are being generated, you have the recombination event taking place in the heavy and the light chain, and you already have the VDJ or VJ combined. Now, it is only in the memory cells that you have the VDJ, VJ, which now get associated with any one of the constant domain. Like I said, in the primary you will always have VDJ C mu C delta. It is upon the generation of memory cells, subsequently the same C mu delta can class switch to any other isotype. Now, when do you see class switching? If one thinks in terms of switching from mu to delta itself, which happens by alternate splicing at the mRNA level, then class switching is remarkably high during the formation of naive cells. You have both. Now, the cells are first, remember, in the ontogeny of B cell development, you will see that IgM is the first immunoglobulin that is expressed on the cell surface, and soon after that also IgD, and there is then you have these double positive cells, so to say, which have both IgM and IgD expressed. Remember always that IgM and IgD here would be identical with respect to the antigen binding. In the secondary response, class switching is remarkably high, where IgM to any one of the classes of immunoglobulins is switching happens. Again, I would like to tell you, which I have specified in my earlier presentation, this class switching requires T and B cell interaction, just like also affinity maturation. All these happen in the memory cells, and therefore, those B cells which are T cell independent, there would be no class switching, there would be only IgM. T B cell interaction, remember, this happens by way of recognition of the CD40 receptor present on the B cell with the CD40 ligand, which is expressed on the T cells upon activation. Mechanism of class switching. Class switching happens by a very well organized class switch recombinase machinery. Now, recombination might ring a bell. We have been talking about RAG1 and RAG2. Now, that is also a part of the recombination machinery. Now, though the VDJ recombination and class switching recombination are similar, there are distinct proteins and enzymes which are required for both these events to take place. While the VDJ recombination is very specific for where recognition of sequences on the DNA takes place, specific sequences, recombination for class switching is a little bit more imprecise, and let us look at what are the events that take place when class switching takes place. First, selection of a target switch region. Now, I have not introduced that, and I will do it in a minute. Selection of a target S region by the recombinase machinery, cleavage of target switch, which would be IgM always, and switch the target S and switch of the mute heavy chain. Now, this happens by one enzyme called AID, which is activation induced deaminase. The third sequence in this event would be after cleavage, repair and ligation of broken DNA ends by NHEJ, that is non-homologous end joining repair system. Again, I would like to say that the joining and cutting in case of the class switch recombinase is not precise, like happens in the VDJ. Let us look at where these switch region genes or gene segments are present. Now, I told you a little while ago that recombination can take place between the C mu and any one of the constant domain barring C delta. Therefore, there has to be a switch region gene on the 5 prime side of every constant domain. Now, some of the constant domains I have not listed here just because of lack of space, but let us look at now the constant domain, the switch region genes which are on the 5 prime side or every constant domain except C delta. Now, let us say in this particular instance, C mu is going to class switch with C gamma 2 B. Therefore, recombination should take place between the switch mu and switch gamma 2b. Looping out takes place. Now, what brings about this looping out we will deal with, but let us just look at the events that are taking place. Now, VDJ, this should be 5 prime. Now, the VDJ, recombined VDJ now is associated primarily with the C mu, C delta, and now needs to undergo class switching to C gamma 2b. Now, the S mu and the S gamma 2b, these two come together, they join, you can see, and this becomes now the rearranged gene again at the DNA level. The rest of the constant domain genes which come after gamma 2b still continue to be present. Now, it is at the transcription level that you will have the VDJ. You can still see the switch, the joined switch regions C mu and C gamma 2b, and the RNA transcript has been formed together with DNA circle. Now, what is this DNA circle? We will come to that in the next slide. Then there is, after RNA processing, you have the mature transcript, and this is going to now synthesize, code for, or give this the message for the protein. Before I come to what are these switch region genes, let us just look at the DNA circle that I talked about in the previous, denoting the previous slide. Now, switch region is seen between the S mu and S gamma 2b on the rearranged DNA, and a part of it which is excised out is seen even as a circle in the cells which are undergoing switch recombination. Now, the joining would take place even in the excised one, just like the switch regions S mu as well as S gamma 2b are a part here, and a part would be here, which is seen in the next slide. Now, these are of course, when they are cut, of course, these would not form circles, but they start, the joining or the enzymes that would ligate would of course not differ or differentiate between the excised one as well as what is in the DNA, and therefore, you will have these circles. Now, let us look at what are the switch region consist of. They vary for different isotypes, but they are approximately 5 to 10 kilobases, each one of these switch region gene segments, but anything which is common between all of them is that they are GC rich. Now, these sequences which are 5 to 10 kilobases of course, also have some motifs, but this switch region genes are, the sequence reads as GAGCT n times followed by GGGGT. Now, this n can be 2 to 5 or n can be 17, as one can see in the switch mu region. Now, obviously, there should be some factors which govern class switching. Are there any factors that govern this? First thing what one can look at is the type of antigen themselves, because during evolution from let us say the jawed fishes to mammals, which are the most complex, and humans absolutely the most complex. In evolution, during this there had to be some kind of development of the heavy chain, so that we become better and better, or in evolution animals became better and better by evading the pathogens or mounting a very well and robust immune response. So, how do these classes of immunoglobulins differ? IgM, IgD, IgE alpha and so on, all these differ in their constant domain genes and therefore the effector functions. It is the VDJ or the variable region genes which recognize the antigen. Now, there of course, the difference would be with respect to the affinity by which these receptors or antibodies bind to the antigen. But it is the effector functions of the rest of the molecule which actually brings about the actual function of these immunoglobulins. One example I can give you is with respect to IgG2A, which is an isotype that is made in response to viruses, and in human this would be IgG2, and the Fc regions of these immunoglobulins can bind to some of the cells of the innate immune system. Now, thereby this would be an antibody driven mechanism where the antibody binds to the pathogen through the Fc receptor. They are brought in close proximity to the cells of the innate immune system and thereby now bringing the pathogen close to, let us say, a macrophage. So, that engulfment is faster, better, more efficient. So, amongst the factors that govern class switching, the type of antigen themselves would be the first. So, you have now an example that I gave you of viruses. The second would be microenvironment of the plasma cells themselves. Example, plasma cells leaving the Peyer's patches or mesenteric lymph nodes exhibit class switching to IgA. IgA is seen mostly in body fluids, and IgA, in fact, if you look at the different immunoglobulins, maximum secretion of any immunoglobulin class would be IgA. We will come to that in the next lecture, but Peyer's patches which are present in the intestine. So, plasma cells which leave that region would class switch to IgA because of the microenvironment. Allergens, in presence of interleukin IL-4, excess interleukin 4 causes switching to IgE. Now, again why? Because IgE brings about mast cell degranulation. Again, this we will deal with in the next class. Another factor that would be governing would be the route of immunization. Administration of an allergen, let us say through the nose or the nasal passage, may give rise to IgE. However, the same allergen when injected intradermally or intramuscularly would give class switching to IgG. Therefore, route of immunization also is one of the factors that governs class switching. Finally, the genetic background of the responder. This I can explain by way of people who are atopic individuals, which means they respond by way of hypersensitivity or allergic reactions. Now, these individuals already have a background, the genetic makeup is such that they respond to allergens by way of class switching to IgE. Now, the same allergens would not evoke such a response in other individuals who are not atopic. We go on now to seeing factors at the molecular level. Are there any factors that are involved in class switching? People have looked for factors such as transcription factors or factors which now in this case would allow the class switch region of the IgE to come in close proximity with the class switch region of mu or IgM, and this interaction would mean that the cell would now allow VDJ recombined region to combine with the constant domain E. All this would happen only if the chromatin reorganization takes place in the switch region genes. So, people have looked at what would be the protein complexes that interact with the switch region or motifs within the switch region. and there are two such candidates that have been discovered called SNP and SNAP. SNP is NF kappa B P50 homodimer and SNAP E2A E47 heterooligomer. Now, what has also been seen is sequences surrounding the double strand breaks, when the switch region, when the cutting takes place by the activation induced deaminase, these double strand breaks contain short inverted repeats that facilitate stem loop structure formation for targeting by the recombinase. This looping out is absolutely essential, as I have shown in the previous slides, and this would mean that there should be some proteins which allow this looping structure, stem loop structure to be formed. There are other proteins which have also been shown to bind to the switch region gene segments, and they are LR1 in case of the switch region for gamma 1, gamma 3 and it should be SA, it should not be CA, this denotes constant, but we are talking about the switch. So, this should be S alpha. There are also those that bind specifically to the switch regions of the mu heavy chain, the sub P2 NFS mu. Now, I would like to emphasize here that though these factors have been seen to bind to these motifs and switch region genes, but none of these have been actually demonstrated to bring about class switching in vitro condition. By DNA binding studies, these molecules have been identified. Now, the other way to look and identify molecules that would bring about class switching would be to identify the DNA binding proteins in the nuclear extracts of cells which are undergoing class switching. And 4 such proteins have been identified, and which actually the functions of which in fact would be required for class switching to take place. The 4 proteins are nucleophobin. This protein is a Recce like DNA loop forming, has Recce like DNA loop forming activity. PARP, which is a polymerase required for DNA repair. So, PARP is poly ADP ribose polymerase. Nucleolin, this is a component of the S mu binding protein LR1, which we referred to in the previous slide, and SWAP70, this is expressed strongly only in B cells and it is involved in switch recombination. The last but not the least, the most important protein in fact, which has been shown to induce class switching is AIT. In fact, this is the only enzyme that has been shown in vitro to induce class switching, and this is what I am going to explain to you. Now, this was a paper that was published in 2002, which showed the importance of activation induced cytidine deaminase, and now one can imagine that this enzyme was shown to bring about class switch in fibroblast cells. Once again, as in some of the previous experiments that I discussed with you, people have this Okazaki et al., they have made an interesting construct which was transfected in fibroblast cells. So, they have the construct where there is a promoter, which next to the promoter is CD8 alpha. Now, CD8 is a receptor, you all might remember from some of the lectures that you have heard so far. CD8 alpha beta, these are the two which form a co-receptor for the T cell antigen receptor. Now, this is membrane bound, therefore, they have included this in the construct. Now, there are 2 switch region genes, those that are for S mu or the heavy chain of IgM, and switch region for alpha. There is a polyadenylation signal which is on the 3 prime side of the C mu. Now, after the C S alpha, the switch region alpha, there is GFP, that is green fluorescence protein, a gene coding for green fluorescence protein. Now, when the cells were first selected, they were transfected with this construct and selected by the thymidine kinase gene, which was the selection marker. And then when these cells containing, all the cells containing this construct were then transfected with AID, the construct that now encoded the gene for activation induced deaminase, which is this enzyme is a 24 kilodalton, which is constitutively expressed in activated B cells. So, when this was transfected in the fibroblast along with this stably transfected construct fibroblast, then the cells started to fluoresce green. How did this happen? That the switch region combined because of the activation induced deaminase, and now the CD8 alpha chain fused with the GFP, and since this is a cell surface expressed protein, the cells that allowed class switching to take place also fluoresced green. This was a rather elegant experiment where AID or AID was shown to induce class switching, and in a system where it does not normally take place. So, the paper also starts to say a few aspects that I will deal with now, that AID induces double strand breaks in the switch sequences. So, though there is no specificity with respect to this double strand break happening, why I keep saying specificity is because remember RAG1 and 2 they recognize specific sequences, the heptamer and the nonmer of the RSS, recognition signal sequences. There are no RSS in the switch regions, but AID recognizes certain motifs in this GC rich repetitive DNA elements that form the switch region and induces double strand breaks. Now, the recruitment of AID, how does this happen to the switch region, is not by the recognition of the switch region by the AID, but there are factors that bind to these regions and in turn bind to or recruit AID to this particular locus. Now, another thing, again activation induced deaminase, as one can imagine, deaminase initiates also diversity of immunoglobulin genes through deamination of cytosine to uracil. Now, this is another factor that governs the variability, but what we are dealing with of course is the constant domain and the class switching. Now, just to summarize this part, in the primary immune response, always the VDJ recombined region associates with IgM, IgD and express on the cell surface. Therefore, in the first immune response that is generated, now this is a naive cell, when such a naive cell encounters an antigen to which it has this receptor, specific receptor, undergoes an activation process, starts to proliferate and differentiate. Now, the immunoglobulin that are synthesized as antibodies by cells originating from this first cell in the primary immune response would always secrete IgM. Now, there are few cells that become memory cells, and these, when they encounter the same antigen again and when they get help from T cells by way of CD40 ligand presentation to the CD40 receptor, these undergo class switching, whereby the VDJ, this very same VDJ now associates with any one of the constant domain gene segments, which is dictated in turn by the type of antigens that have been introduced. I would like to say here, though I keep saying VDJ, which is already recombined and it is identical, remember from the previous lecture, that memory cells also express error prone polymerase, DNA polymerase, which can bring about mutations and increase in the affinity of these immunoglobulin antigen binding genes. So, now the class switching happens so that the immune system gets better and better at eradicating almost all the type of pathogens or soluble factors which are foreign to us. Therefore, in the next class, it will be easy for you to recognize why do we make IgM, why do we make IgG, the different classes of IgG, subclasses of IgG, or IgE or IgA. Now, let us go back to looking at the regulation of the immunoglobulin genes. Now, let us go back to seeing why RAG1 and 2 are expressed only in lymphoid cells. There has to be something that regulates the RAG1 and 2 expression. So, when people started to look for factors in the T and B cells, which now allow expression of RAG1 and 2, they found a gene called Ikaros. Ikaros, like I said a minute ago, is highly lymphoid cell specific. Ikaros codes for transcription factors. These transcription factors, they encode 5 zinc finger binding proteins, and this is by alternate splicing. Now, the targets for the Ikaros gene products primarily RAC proteins 1 and 2, terminal deoxyribosyl transferase. Remember, these are the one, this enzyme adds N nucleotides to the cut ends of the hairpin structure that is formed once again by RAC proteins. The targets for Ikaros is also immunoglobulin heavy chain and immunoglobulin light chain. Another target is MBL, immunoglobulin alpha. Remember, this is the immunoglobulin alpha, which is present as a co-receptor. Sorry, this should not be there, not surrogate light chain, but the co-receptor for the antigen receptor. Okay. Also members of the CD3 complex. CD3 complex, you have already studied the antigen receptor in case of the T cells, and you know that CD3 is the co-receptors associated with the TCR. Mutations in the Ikaros gene results in abrogation of the lymphoid cell development. Therefore, one can imagine that not only RAG1 and 2, but RAG1 and 2 themselves come under the control of Ikaros gene, and therefore, mutations of this gene itself will abrogate the lymphoid cell development totally. Let us look at the regulation of transcription of the now recombined immunoglobulin genes. There are 3 major regulators, like the same in almost every gene. There are promoters, enhancers and silencers. Promoters, I mean all of you all would know already, promoters are approximately 200 base pair sequences which are upstream of initiation site and AT rich data box, which is the initiation site. Enhancer, these are sequences that activate transcription from the promoter sequence, and they could work in cis and trans. Silencers, which downregulate transcription. So, you have enhancers and silencers, and the names themselves denote that. The silencer also downregulates transcription, both cis and trans. If you look at the heavy chain, this is what I wanted to show you, that the promoter, there has to be a promoter on the 5 prime side of every leader sequence, which is on the 5 prime side of every V gene segment. So, you can see the presence of promoter, and every variable gene segment, which is a part of the variable domain of the immunoglobulin gene, has a promoter, not only in the heavy chain, but you can see also in the light chain. Now, there are enhancers which are on the 5 prime side of the constant domain, on the constant domain or the 5 prime side of the constant domain also in the kappa and the lambda light chain. In the germline DNA, the promoter and the enhancers are 250 to 300 kilobase away, and rearrangement of any one of the V with any one of the J and D would bring these close together, because of which transcription is influenced. Are there any specific motifs present in the promoter region of the immunoglobulin genes? Yes, there are octamer motifs which are highly conserved, and one can see the motif ATTGCAT in the lambda and kappa genes, the light chain genes, whereas in the heavy chain promoters it is ATGCAAT. This is interesting. These sequences are very conserved in all immunoglobulin genes on the V segment. Something I would like to again introduce to you, that this phenomenon of every B cell which is already committed to look at one type of or recognize one type of antigen. Now, recombination happens only in one of the alleles, never on both alleles at the same time, and I am going to deal with this once more, but there is a coordinated way by which recombination takes place. It is known that the immunoglobulin heavy and light chain genes of only one parent chromosomes are expressed per B cell. The other one is excluded. Now, allelic exclusion, this allows or ensures that a single B chain will be specific for a given epitope. The selection of which allele of each pair is rearranged is totally random. We do, all of us do get both paternal and maternal alleles, and they are equal. However, in the B cells, which heavy chain allele is now taken or rearranged first or is successful is totally random. But this is important, so that one B cell recognizes only one epitope of any antigen, and therefore, makes antibodies of only one type. Now, regulation of expression of immunoglobulin genes happens at the DNA level by several factors, but it is not very easy to talk about all of them. I would like to talk about those which are very important. For example, ID2. Now, these are more recent literature, and you know that immunology in fact is young science, and there are more and more additions to the information that we already have at regular intervals. So, regulation of expression of immunoglobulins can happen at the DNA level as well as at the protein level. I will give you examples of this. At the DNA level, now one should have minimal IgE type of an immune response. Now, where would one want an IgE type of immune response? For example, in case of pathogens which are large, let us say helminths, worms that enter our system, where the macrophages cannot engulf, neither will the antibodies be able to kill. However, there needs to be some factors which should now eradicate these worms, and the immune system is primed to do that. Is the IgE antibodies that actually participate in this? And I like to say that, now let us say one is infested with worms, then there are factors which allow the class switching to take place between IgM to IgE. Now, this happens through transcription factors called E2A, which bind to the switch region of IgE, making that available now to undergo class switching. Now, in case of the pathogen like helminth, it is required that the immune system makes IgE type of antibodies, because these antibodies, as soon as they are secreted, they are bound to the mast cells. When the antigen is recognized, molecules from the antigen are recognized by these IgE, which are sitting on the mast cells, cross linking of these receptors take place. Thereby, there is now secretion of a large granules which are already present in the mast cells, preformed granules. The preformed granules now attack the helminth. No cells can engulf these helminths because they are too large, not like bacteria which can be phagocytosed. So, there is an arsenal of molecules that are now thrown on the parasite, on the extracellular parasite. Now, all this happens only because of the presence of IgE, which is made in response to the pathogen, and now the IgE is sitting on the Fc receptors which are specific for IgE on mast cells. Now, to prevent such switching to take place between IgM and IgE for all antigens, there are transcription factors like E2A which are down regulated by ID2. Now, these ID2 are present all the time, so that now they suppress E2A and therefore suppress the class switching from IgM to IgE. Now, this is happening at the DNA level. Now, you can have regulation of expression of immunoglobulin by way of antibody production. So, you inhibit the cells from proliferating, differentiating to antibody producing cells at the protein level, and this can happen by, let us say, receptors like CD22, which I have not talked about before. CD22 is present on surface of B cells. CD22 is a 140 kilodalton protein. It is a glycoprotein in fact. Now, this has in the intracellular domain ITIM. Does this ring a bell? Remember ITAMs and ITIMs. When I was talking about signaling in B cells, I talked about ITAMs. ITAMs are immunoreceptor tyrosine based activation motive, and ITIM immunoreceptor tyrosine based inhibitory motive. Now, both ITAM and ITIM do get regulated or get activated by way of phosphorylation, but while ITAM now recruits kinases, ITIM recruits phosphatase. So, that is what CD22 does. But how does it do that? Interesting that the cytoplasmic tail of IgG, again I hope you remember this, that the cytoplasmic region of the IgG has 28 amino acids as opposed to only 3 amino acids of IgM and IgD. Now, what does the cytoplasmic tail do? It does not have ITAMs. Remember immunoglobulin, the antigen receptor itself does not have any motives. But what does this 28 amino acid do is it inhibits or it interacts with the CD22 phosphatase molecule intracellularly and inhibits the activation of the CD22, thereby inhibiting the ITIM and thereby inhibiting the phosphatase, which would mean that a B cell that is making IgM and a B cell, another B cell which is making IgG, it has been seen that always the IgM produced by a cell is much lesser in the number of molecules made as compared to IgG. So, there is regulation by inhibiting kinases. Does this remind you of another molecule which I talked about in reference to signaling? Yes, you are right. This is Fc gamma R2. Sorry, I missed out B. It should be Fc gamma R2b, which is a receptor which is present on all B cells. Remember this, that when the immune response to a particular antigen is very high and you have now circulating antibodies present. The circulating antibodies, when bound to the pathogen, let us say, by this, now the circulating antibody can now bind to Fc gamma R2b and co-ligation takes place because another epitope recognizes or is recognized by the antigen receptor of the same B cell. This brings about a conformational alteration in the Fc gamma R2b and this undergoes phosphorylation for recruiting phosphatase, which would now start lowering the activation index of that particular B cell, in turn inhibiting the immune response. So, there are a score of different molecules both at the DNA level as at the protein level, which ensures that the antigen receptor mediated activation of B cells and bringing about now activation of the or synthesis of antibody producing plasma cells can be modulated such that immune response does not go haywire. Now, the regulation really in total would be also at the level of the recombination event itself. Exactly how this happens is not very well known, but as I go along I will tell you where this recombination can be seen to be regulated, primarily at the progenitor B cell, which undergoes recombination at the heavy chain gene first. Heavy chain gene at the variable region, recombination takes place first between any one of the D with any one of the J. Remember, this is important for any one of D, J, then recruiting any one of the V. Now, if the recombination is successful, then the cell goes to recruiting any one of the V. A productive allele 1, then if this happens, now the products of this allele would inhibit recombination which is happening, which can happen in the second allele. So, there is regulation over here. Now, it is believed that the signal joins that are formed when VDJ recombination is taking place, these inhibit the allele 2 in this particular cell from undergoing recombination. In case this recombination fails, only then the second allele starts the recombination process. Interestingly, only when VDJ recombination has taken place and the heavy chain transcription and translation happens, this can associate with, remember, the surrogate light chain. The surrogate light chain itself cannot recognize an antigen, but can associate with the heavy chain such that surface expression takes place. Now, this surface expression is the clue for the recombination of the light chain genes to start. The surface expression also inhibits the second allele of the heavy chain from starting. So, that means regulation at yet a second level. Now, always recombination in the light chain gene starts with the kappa light chain gene assembly, allele 1, and if that does not happen, if there is non-productive allele 1, then the allele 2. If both the alleles of the kappa light chain genes fail to reorganize themselves, then allele 1 of the lambda light chain is recruited, and if that does not happen, then allele 2. But remember, all along it is a very coordinated process. At no point of time does recombination of the heavy and the light chain occur simultaneously, nor the kappa and lambda happen simultaneously. This ensures that one of the alleles only of the heavy and any one of the light chains kappa or lambda recombine, and now you have an immunoglobulin molecule on the B cell surface which has the capacity to recognize only one type of antigen or epitope. So, with this I will end the lecture here. My next lecture deals with the structure of all the immunoglobulin classes and the effector functions. I like to keep saying this, that during the course of the course of evolution, we have now the immunoglobulin genes, which in the first the vertebrate that it appeared was only IgM, and subsequently the other classes of immunoglobulin started appearing, and we have now in the finest form of the immunoglobulin classes by way of 5 different classes of immunoglobulin, each of them with specific function. Thank you.